Well, the forecast looks relatively sunny and clear for a while, so you should have a nice spring break, depending upon where you're going next week. Couple of orders of business. Did everybody get my BB Learn announcement today, just about what's happening on Wednesday? So I pre-recorded Wednesday's lecture. As I had a trip planned about a year ago with one of my daughters, and so we leave tomorrow for New Zealand. And it just so happens that we had booked that trip before I signed up to teach this semester, so that's the one. Other than snow, you guys are like, oh, it's no big deal. We haven't seen you because of snow, so have fun in New Zealand. So hopefully you're thinking that. But Lecture 3 has already been recorded on YouTube, and it's posted. It's ready to go. And the nomenclature is the S23, which stands for Spring of 23. I know, it's kind of crazy. But that's how I've labeled this year's lectures. The old lectures are still there, and you're welcome to look at them, but content from this semester is S23. Now, Wednesday, I know all of you are available, and this room is available. And so we're going to have a review session that the TAs and the SIs have decided to do in this room on Wednesday at the regular time. Okay? I highly encourage you to do this. I'm getting a lot of emails, and it sounds something like this. Dr. Keller, I should have reached out sooner. Really enjoying the class. However, my grade is not reflecting where I think it needs to be or where I think I want it to be. And I'd like to meet with you and talk a little bit about my exam too, and also about what I need to do differently. So guess what? Wednesday is something different. You should show up. It's something that can help you. And just some data. The SI sessions are averaging about 5 to 15 people in attendance. 350 of you. And I know you're telling me, Dr. Keller, we're busy. We got a lot of things going on, and it just doesn't work. Well, there's more than one slot. And then the other one is the TA sessions. Like the day before the exam, I know it was a Sunday, and it was like at 1 o'clock or noon, but it was on Zoom. It's actually recorded as well. There were only about 40 out of the 350 that showed up. I think 40 is pretty good. But I would like to see hundreds of attendance. And so if you're going to ask me, hey, what could I be doing differently? That's different. If you're not going to the SI sessions, you're not going to the review sessions, you're not looking at the study guides, you're coming to lecture or you're watching the lectures at home, you're not even coming here because you know I record them. It's no secret. You can figure that out first week of school. But newsflash, this class requires you to spend a lot of time with the material over and over and over again. In the Stone Age, we called that technique studying, but we have to rebrand everything, so now we just call it spending time with the material. Okay? You need to study. And we're creating opportunities for you to study. And so if you're going to ask me, hey, what can I be doing differently? These are some of the things I'm going to tell you. I'm going to start with, are you coming to class? Well, why do I need to come to class? You record your lectures, and I watch it on YouTube anyways. Well, if you watch it one time, it's not as good as rewatching it with your notes, coming the first time, taking notes, rewatching the lecture, going back over the stuff that you fell asleep on or you weren't paying attention, and you fill in the gaps. That's much better. So let me ask, do y'all think do you think the lectures are pretty okay? Are they pretty organized? It's not like I'm having to Google, like, oh crap, I don't know what's on the next slide. Right? I'm relatively prepared. Do you think it's the first time I've done the lecture when I show up here and see you all? So if you want to do better in this class, you're going to have to do something different. Midterm grades are going to be posted. I know you're going to freak out, but the whole point of that is there's time to make adjustments. All right, some stats. It's no surprise that we've got sort of these bonus opportunities in front of exams, right? So you guys see how I do this. Instead of curving the class at the end of the semester where you don't know what's going on, I create these bonus opportunities where you could take advantage of doing some extra work. You read a peer-reviewed paper that one of your classmates suggested. They didn't know they were going to suggest it, but it was on fish skin and wound healing. I thought it was a great topic. It came from over here. I don't even know who it was. Don't remember. But I looked it up, and I remember seeing a paper on it years ago, and I thought, hey, that would be a great topic. It's relevant to what we're doing. You read the paper. You answer some questions about it. You're in the peer-reviewed literature. That is not a 200-level class activity. That's a higher-level class activity. I'm willing to hand out or award bonus opportunity points, and many of you on Exam 2 leveraged the heck out of that, which is great. But there were 14 possible points. The average was 12 out of 14. Okay? If you take that and you add it to the exam score, the average on Exam 2 was a 71%. And that's what you should do. You should take your bonus opportunity and add it to your Exam 2 score, and that's your Exam 2 overall score. You with me? You do the same thing with Exam 1. So I am adjusting where along the way so you know more real time how you're doing in this class. Make sense? Nobody can predict the future, but a lot of people say look at the past. It's an indication of what's going to happen in the future. Hint, hint. Okay? 71% average if you take the combo. Bonus opportunity plus the exam average. So you can do the math and figure out that the overall average was relatively low. So the bonus opportunities are necessary. Of the students, you're like, you're watching us? Of the students that came to the review session, plus the bonus opportunity, the average was a 79.5. Bonus opportunity plus the exam. That rounds up to a B, 79.5. You look at the syllabus. So it's a whole letter grade better for those students that are attending review sessions. You're like, well, wait a second, Dr. Keller, the data is inconclusive. You can't tell me that if you go to the review session, you're going to do better on the exam. That is a true statement. I'm saying the students that went to the review session ended up doing better on the exam. It's maybe not a cause and effect, it's a correlation, but my point is, be that group. Be the group that does better on the exam. Do everything that they do. That's why I want to see, instead of 40 at review sessions, I want to see 150. I'd love to see 350 of you. I just know it's unrealistic. Make sense? Is that information? Let it sink in. Next week is spring break. So you know Wednesday you have an opportunity to wow me. I won't even be here, but I'm going to hear it from the TAs and SIs of what our attendance looks like. And I'm expecting a lot of you here. I'm expecting all of you here. Because I think that's something, you have the time, it's in your schedule, and you should show up and take advantage of that opportunity to talk over nerve physiology review on Wednesday. Okay? And it's not a substitution. I already recorded the lecture. That's not like I'm getting out of my job. I already took the time and did the lecture, so you have my lecture, but this is in addition to it. It'll help augment what you're learning. Fair? Make sense? Hang on, let me silence this. Okay. Spring break. I think it's important that we take time off. I like to work hard, play hard. Over spring break, there are no assignments. There's no lectures, there's no extra readings. Okay? There is no quiz. Your next quiz actually doesn't show up until after we get back from spring break on the 26th, which is the day before, Sunday before the next exam. So you don't have any assignments from me next week. I don't know what your other classes are requiring, but it is a great opportunity to have a little relaxed time, have a little fun, but maybe don't get stupid. Like, don't lose everything you've already learned. I would highly recommend you carve out a little bit of time to go over nerve physiology and refresh and stay relevant. So you don't come back after spring break and be like, oh my gosh, I don't know anything. Is that fair? Okay. I'm not saying you have to do an assignment. I'm just trying to encourage you not to regress over spring break. Go have fun. Go relax. Be safe, please. Come back to me. But don't totally blow off the week. It's a great opportunity. You should be able to get internet most anywhere you go. You can rewatch a lecture. You know you can speed those lectures up, right? Or you can slow them down. You can go 1.25 or you can go 0.75. I'm like talking like this. Okay, so you can dial it in however you like. You can close caption it if you're on an airplane, you don't have great headphones. Okay, let's move on. Lecture 2, before we go there. Who earns a living driving people away. Hopefully I haven't driven you away today. What's that? I heard it. A taxi cab driver. Yeah, today in 2023 we say an Uber driver, but yes, taxi cab driver. What do you get when you cross an elephant with a fish? Actually, elephant spelled with an E. Elefante. My daughter wrote it. With a fish. An elephant, or elefante as she spells it, with a fish. This is apropos for spring break. Swimming trunks. Okay. Why was Tigger looking in the toilet? What's that? He was looking for poo. All right. My girls, when they were young, really liked that one. They thought that one was just too funny. Let's talk second lecture, nerve physiology. And I'm going to tell you the third lecture on nerve physiology, there's not a lot of new material in Wednesday's lecture. It's kind of reinforcing what we cover today. Okay? And you're going to be pausing it, you're going to be stopping, going, you're going to be taking out a piece of paper, you're going to be drawing stuff, labeling it. So it would be best if you had scratch paper and you're in a place where you can actually write some stuff out for Wednesday's lecture, because it's kind of trying to review what we're going to cover today. So nerve physiology, we're going to look at action potentials. And what I love about this slide, and this was a few years ago, this wasn't Snowmageddon here this year, but this was another big year that we had. And I think you see this in biology. You see this throughout the disciplines within biology. There's a lot of conservation of architecture. And I actually think that these aspen trees look like neurons. So whether you're going somewhere warm or whether you're going somewhere cold next week, you might see some things in nature. In New Zealand, we're going to kind of be remote, hiking around, and I'm excited about that. It's fall going into winter there, southern hemisphere. So we probably won't see trees that have lost their leaves all the way, but maybe. We'll see. We'll find out. I've never been there, so I'll report back. But the architecture is what I wanted to point out to you. I want you to start to kind of appreciate the way the neuron just looks. And maybe when you just look out in landscaping here locally, you might, like me, maybe you're a nerd, you might say, hey, that aspen kind of looks like a neuron, right? And those look like dendrites that are collecting the signal. And these nerves, the whole point of the nerve is to actually bring in a signal, move the signal somewhere, integrate it, and then send it out to do something, a response. And that was what we talked about in our first lecture. So I want you to take a minute here, scratch a piece of paper or on your tablet, your pad, your iPad, draw a typical cell. This somatic cell throws people off. That's just another name for body, like a typical cell in the body that's at resting membrane potential, and label where potassium, sodium, and chloride are inside and outside the cell. So if you're graphically challenged, just draw a circle, put a small little circle inside of it, color it in, that's the nucleus. Outside of the big circle is the extracellular fluid. Inside of the circle is the intracellular fluid. Put K plus, Na plus, Cl minus in triplicate, or in a grouping outside, and the same thing inside the cell. And then tell me high, low, high, low. So if it was confusing, I meant for you to do it now, just in case you're curious. Or attempt. This is what lecture 1. We cover this in lecture 1, in case you're curious. How we doing? We'll do a lot of this in lecture 3, a lot of this sort of interaction kind of stuff. You'll be doing it at home, on the ski lift, on the beach, wherever you're going to be. I actually think this would be a great tool for the exam, is to do these drawings on your test packet. Draw them out. Find a blank area. Write it out so you don't forget it. You ready? So it should look something like this. I expect, shadings and three-dimensional structure. No, I'm just kidding. But your extracellular fluid up top, your intracellular fluid down low. If you drew a circle, your extracellular fluid is outside the circle, your intracellular fluid is inside the circle. Conceptually, you can get the same things across. Sodium is high outside the cell. Do you need to memorize it's 145 milliequivalent per liter outside the cell on average? Not really. I want you to memorize or remember that it's high outside the cell. It's high in relationship to inside the cell where it's low. Is it okay if you memorize these numbers? Yeah, for sure. But I'm more interested in you memorizing sodium concentration is high outside the cell and it's low inside the cell. Potassium is the opposite. It's high inside the cell and it's low outside the cell. Chloride ion, high outside the cell, low inside the cell. And I think this foundational piece that we talked about in the last lecture is going to be really important to you. And the way we're at the membrane of the nerves, does everybody understand this? If we boil down now to a nerve, not just any body cell, we boil down to a nerve, we're at the nerve neurolemma, the membrane of the nerve cell. In order to get an electrical current to move, we're going to move charged ions. And you're going to move charged ions in one little area, and then the next little area next to it, the next little area next to it, so on and so forth, really fast. In the area of that direction, you're not going to move ions, so the electrical signal is going to go this way. You with me? So it's like squeezing toothpaste according to the instructions. Like, what? Who knows what I'm talking about? Okay, any takers? If you turn over the tube of toothpaste, it says for best results, squeeze from the bottom up. So if you squeeze from the end and then you squeeze this way, you systematically move the toothpaste in one direction. Some of you are like, just squeeze in the middle, man, and fix it later. Okay? If you squeeze in the middle, the toothpaste is literally going both directions. It's got an opening on one side. So with this type of movement of ions, it's systematic in one direction, and we'll look at that today. Okay, look at the cell that you drew, or fix it. I want you to pair up, person A, person B, next to you or behind you or in front of you. Person A needs to explain how voltage gates and ligand gates are moving ions in and out of the cell. Person B needs to explain how channels and mechanical gates are operating to move ions in and out of the cell. Ready? Try it. You're like, I don't know if I can. Just try. Practice. Okay, let me bring you back in. If this is really difficult for you to do, you could retry it on Wednesday as a group activity, just giving the TAs and SIs some ideas. Okay, walk away and say, okay, now I understand what you want me to do. Let's hear from some folks that maybe have an idea of, say, voltage gate. How does a voltage gate work? What does it do? Maybe give me an example. Any takers? Okay? It is. A voltage gate is triggered by an electrical charge, or let me substitute a different word. an electrical potential. An electrical potential. If you did the reading, you might see that we kind of refer to these as potentials. Like, what is that? In electrical circuitry, a potential means a difference in charge. So a voltage gate is triggered by a difference in charge or a potential. And then it allows for an ion to move through it. That's why it's called a gate. Two great examples, sodium and calcium voltage gates. Will move these ions when they see an electrical potential that causes a trigger. It's like you walk up to, like a fancy house, or you drive up to a fancy house, and you pull up, and there's a camera, and you hear this bzzz, and then the gate opens. And you're like, wow. Okay? Same concept. Ligand gates. Any takers on explaining how does a ligand gate work? What is it? What does it do? Give me an example of it, or maybe just an example. Maybe you don't know how it works, but you know an example. Yeah, that's exactly right. That's knowing a lot right there. She said, doesn't something have to bind to it to allow it to open? The answer is yes. You call that a ligand. A ligand binds. Now the gate's going to open, and it's going to allow ions to flow. A perfect example of this is the neurotransmitter acetylcholine. Okay? Bless you. Acetylcholine binds to its receptor on the postsynaptic side and opens up sodium gates to allow sodium to flood into the postsynaptic side. Okay? All right, let's go to channels. How do channels work? What are they? Maybe what's an example? Any takers? This may not be as obvious. Okay. ATPase pump? Yeah. So I'm going to say yes, but I'm going to modify and say you perfectly described a mechanical gate. Okay? So a mechanical gate, we'll skip to there and then I'll back up to channels. Like I said, they're not as intuitive. A mechanical gate, they open or close in response to some physical distortion of the membrane surface, sensory receptors, pressure, or touch. And so they were going to trigger opening based on mechanics. Something triggers them to open or close and moves ions one direction or another. Now with channels are ones that are always open. A channel is always open. It doesn't really close. Like leak channels, for example. So a great example here would be a potassium leak channel. Okay, with this information in mind, I want you to answer this question. Which is in higher concentration inside a typical cell, body cell, or a somatic cell? What's in higher concentration? Sodium, potassium, calcium, chloride ion, or two of these? If you know the answer, raise your hand. If you want to shout it out and you're raising your hand, go ahead. Potassium, B. Okay? These questions that I'm asking are fundamental concepts of resting membrane potential that we need to understand before we move on. Because in the second half of this lecture, the pace is really going to pick up. I'm kind of testing to see if you already understand Lecture 1. Right? It was an online YouTube lecture. I understand that because of the snow and all that. But it's important that you're familiar with it. Okay, let's tackle this one. What causes a cell to be negative? What causes the cell to be negative? Presence of chloride ion inside the cell. Presence of chloride ion outside the cell. Presence of nucleic acids inside the cell. Presence of proteins inside the cell, or two of these. How many of you like A? B? C? D? Or E? Oh, wow. Okay, so maybe it's a hung jury. So two of these, which two are they? nucleic acids and proteins. So if you said C or D, you were half correct. So nucleic acids and proteins, mRNA, DNA, proteins, nucleic acids, they're all slightly negative charged. And they help to maintain or actually kind of keep the basal level of the cell inside being slightly negative. The sodium-potassium ATPase helps to maintain the negative environment inside the cell because it pumps 3 sodiums out for 2 potassiums that it brings in, right? So it helps to maintain that negative charge. But what really kind of establishes it is the fact that you've got negative anions inside the cell. All right, let's watch this video.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from negative 40 to negative 90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If the potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential.
Okay, that's all I want to show on that. All right. We're going to go through a systematic view of the stages of what that little clip showed. Okay? But first off, answer these couple of questions. The resting membrane potential of a cell is due mainly to what? So another contributing factor, I'm just trying to get from you, what are the contributing factors that make the inside of the cell slightly negative? Open sodium channels, open potassium channels, saltatory conduction. Some of you are like, I have no idea what that word means. Differences in calcium concentrations across the membrane. Anybody want to take a stab at it and explain? Looking for a volunteer. The open potassium channels. The answer is correct. Why? Okay, that was really well said. So I'm actually not even going to repeat it. I'm going to have you say it in the mic. Why is it B? So which way is potassium going if it's leaking? Potassium's leaking out of the cell. Why is it leaking out of the cell? It's high where? It's high inside the cell, so it's going to move down its diffusion gradient and leave the cell if it's got a channel. This is a Unit 1 principle of diffusion, right? So this is where it's really important. Unit 1 stuff. I know it was boring, and you guys were like, why? Why do we have to do this again? We're moving ions now, and if you don't remember this stuff, it's really difficult to synthesize it. And it's so much easier to remember it if you understand it. If you understand why, it's so much easier than just memorizing it. Okay, so let's look at this table. Fill this table out for me, or if you've got a scratch piece of paper, make a box, like 4 quadrants, 1, 2, 3, and 4. Or on your tablet, your iPad, or your printed notes, you're going to either answer, is the ion sodium diffusional force, this first box, is the diffusional force into or out of the cell? Is the electrical force of sodium based on its charge, is it into or out of the cell? Is the diffusional concentration or diffusional force of potassium into or out of? So you're putting into or you're putting out of in these boxes. And this is the information that came off of your figure that you drew, that I re-showed to you out of a textbook that had the blue and the orange transmembrane proteins. You understand the assignment I'm asking? So do that right now. And we'll listen to the wisdom that was over here and build on that. So what's our first box? Sodium, diffusional concentration forces, into or out of? So the way that we're thinking about this is if you open a gate, whatever gate it is, a voltage gate, at rest, given this information, the extracellular fluid is high, the intracellular fluid is low, diffusionally, which way does it want to go? Into the cell. Thank you. That's the diffusional force, down its concentration gradient. What about the electrical force? Is it into or out of the cell? If the sodium is positively charged, the inside of the cell is slightly negative. Positive likes to do what? Is attracted to a negative spot, so its electrical force is into. Does that make sense? How about this? How about potassium? You open a gate. We just had the answer over here. Is the diffusional force of potassium into or out of the cell? Out of. How about the electrical force? Into or out of? Yeah, go with what you think. It's into, isn't it? Yeah, it is. Because potassium is positively charged, it's leaving the cell, but electrically the cell's negative. So there must be a high concentration of potassium that overrides the electrical gradient to push it out of the cell. You with me? Another way to think about this is you've got two forces of sodium when you open a voltage gate. If we have a resting membrane potential, we have high sodium out of the cell, we have a negative environment inside the cell, you open a voltage gate, allow sodium to move, it's got two forces that pull it into the cell. It moves fast, very fast. Into. It's got two boxes that move it into the cell. When you rest and repolarize the membrane and you open a potassium channel, well, it's a little slower because it only has its concentration to move down. It's kind of working against the electrical side of the equation. Does that make sense? Okay. So let's talk about action potentials specifically. These are a terminology that confuses the heck out of students. An action potential is, the word potential means you've got a difference in charge. It's an action potential because that difference in charge is moving, like action-oriented. It's moving down the membrane, like squeezing toothpaste from like hand over hand. Or let's say you have like a big hose from like your pool. You're draining your pool, okay, down in the valley, and you're trying to get the last little water out of the pool hose. Well, you're going to pick it up on one end and you kind of squeeze it and you let it run that direction. Action potentials are charge differences that move. They only occur in areas that have what? A high concentration of voltage-regulated gates. And that happens in the trigger zone, known as the axon hillock, or a portion of the axon hillock. And local potentials, or what we call graded potentials, that are being signified by this staircase of excitatory postsynaptic potentials that we talked about in Lecture 1. Remember, they could be inhibitory or excitatory. These are excitatory, so they're trying to do something, and they build on top of each other. So we're going to temporally summate here. And once you hit threshold, now you get your action potential. And on this side, does it surprise you that it happens really fast if I tell you they're voltage-gated sodium channels? And we've got two forces that throw it into the cell. And that's why it goes up so fast and so quickly. And then over on this side, on the repolarization, this is depolarization on this peak. On the backside coming back down is repolarization. Depolarizing, you've undone the polarization. It's polarized, meaning it's got a charge difference. It's a potential. It has a difference in electrical charge. A depolarization is you make it less polarized. So you're making it more negative. Repolarization is you're repolarizing it. Think about like you're divided on a concept, right? Hey, you got some political topic and you don't agree. You're polarized on the issue. Similar kind of use. But here we're talking about electricity. You with me? All right, let's walk through the 7 steps of action potential success. That's going to be a textbook I write. Just kidding. Nobody will buy it. 7 steps. Okay, so step number 1. Where are we? We're in the local potential. We're in an area that can still be reversible because we haven't hit threshold. So you build here in 1, sodium ions arrive at the axon hillock in the trigger zone. They depolarize the membrane. 2 is threshold at minus 55, right about minus 55 millivolts. 3 is this depolarization event where you're opening sodium fast gate channels. They're electrically sensitive. They get the electrical charge difference. They open the gate. Which way does sodium go? Into or out of the cell? Into the cell. It rushes into the cell. As it rushes into the cell becomes quickly less negative because sodium's bringing a positive charge. That's why you go towards the positive side of this graph. You become out of the negative, cross the 0, and you go to plus 35, in some cases up to plus 40, some neurons. We'll talk about that. But for purposes of today, just look at positive 35. So does that make sense that sodium comes in, it makes it less negative or it makes it more positive? You depolarize that. That's why this membrane potential is positive, because sodium is a positive +1, right? You guys following all that? I know I'm like beating a dead horse here, but I just want to make sure you're getting it. Now what do you think happens at 4? Do you think those sodium voltage gates close? It's a good guess. Because now all of a sudden we're going in the other direction, like an abrupt U-turn. Well, we also have at 3, we have slow voltage potassium gates that open, but they just take their time to open. So the potassium gates open here. Here's where the sodium gates close. 5 on the next slide. 5 is our repolarization event. That's where the sodium voltage gates are inactive and closed, and these potassium gates that opened over here in 3 are still open. They actually take their time closing. They don't really close until 6. And because when they open, which way does potassium go? It goes out of the cell. Exactly. Does it go quickly out of the cell relative to sodium? No, it goes slower. So that's why this rebound is slower than this aggressive spike, because it's going down its concentration gradient, but right here it's leaving the cell, taking a positive charge with it, so it makes the cell more negative, and as it closes slowly, you overshoot. You hyperpolarize the membrane, making it more negative than minus 70, until you reestablish resting membrane potential at step 7. And in this phase right here, what's interesting is you've prevented the side of the axial lemma behind where the action potential is trying to go. It's moving in this direction. So behind that, it's actually more negative, more difficult to trigger an action potential, so it won't go this way. It actually propagates in the forward direction. So the hyperpolarization sort of ensures that you propagate the signal in the forward direction versus squeezing the toothpaste. And imagine you have toothpaste, you have a hole in the bottom of the toothpaste tube. You squeeze in the middle, you're going to get toothpaste going in both directions. So this ensures that you don't get the electrical signal going backwards. Clear? Characteristics of this. Now, actually, let me pause here for a second. Before I continue on, it probably seems like I'm going rapid over that. Okay, but we built in Lecture 1 the foundations to go to the action potential in Lecture 2. In Lecture 3, in the beginning part, I'm just going to have you iterate this. You'll see. You're going to draw it. You're going to label it. You're going to number it. You're going to talk about channels again, what we just talked over. This is the mechanistic components of how action potentials happen. I hope that's clear. And the great thing about it is if you need to re-see it, re-watch it, re-listen to it, you can. Speed it up, slow it down. Okay? Let me ask this question, or let me pose these questions. The characteristics of these action potentials is they're really an all or nothing. Once you hit threshold, it goes. It's full send. Up until threshold, you can change your mind. It can not hit threshold and not fire. But once it hits threshold, it fires. That's the point of no return. They do not dissipate. They're non-decremental. This is important. Once you fire, you don't want it to wear out. Now, in some diseases like MS, you'll have dissipation of the signal. Multiple sclerosis. We'll talk about that here in a minute. These are also action potentials in healthy individuals. They're all or nothing. They do not dissipate or wear out, and they're irreversible. You can't bring it back. You sent the text, it's over. You can't send it, you can't recall it. If we look at some other characteristics of what happens with these action potentials, we've got refractory periods that I talked about. We have an absolute and we have a relative. Now, in the absolute refractory period, you've got no new action potential possible. That's when it's firing, and in the early stages of repolarization. In the hyperpolarization phase, it's a relative refractory period where it can still fire, but the stimulus would have to be much larger than normal in order for it to fire. Again, you don't want to disrupt in absolute anything happening with the action potential because you've decided it's important for me to send this signal down this nerve to go to this muscle to contract and run away from that bear. Right? Or we need to send the signal to this gland to contract, to release the enzyme so I can digest the meal that I just had. Because you get so into the weeds of how an action potential works, sometimes it's like, well, what the heck? Why are we even talking about this? What does this even do for me? This is how you send a nerve signal to a gland or a muscle, to a tissue or an organ to do something. I want you to fill out this table where you're comparing and contrasting an action potential in this column to a graded or a local potential in the far right column. And I'm asking about some characteristics, like is it an all or nothing event? Is it decremental over time? Can it be inhibitory? Is it reversible? Is it short-lived or long-lived? Does it vary in strength depending upon the strength of the stimulus? Where does it occur? And the types of answers that you're going to come up with is going to be yes or no for most of them, except for a couple of little short answers, right? Like where and maybe duration. So these boxes you could fill in with, so duration, is it short-lived or long-lived, as well as where might be some short answers. But everything else is going to be no or yes. And these answers will post tonight at 7 p.m. on BB Learn on an announcement. Okay? So if you don't want to know the answer, don't look at the 7 o'clock posting. It's already scheduled because I'm leaving on a flight, so I wanted to do it before so I don't forget. If you want to do it after class, great. If you want to wait until tomorrow or Wednesday, fine. But your answers will be on BB Learn in the announcements. Fair? Understood? All right, switching gears a tad. Let's talk about some nerve physiology as we propagate signals, and we look at some other features. So I talk about this tube of toothpaste. And some of you are like, I'm so confused about this tube of toothpaste. So let's do this. Let's look at, here's the cell body. Here are the dendrites. This is where we're receiving the signal, and the signal is moving from left to right. You can see the arrow. Here is the axon, and then here is where the axon terminates into a gap of a synapse to a postsynaptic membrane on another nerve or on a muscle. Eventually, you're going to start getting into muscles in lab, okay, this week. I like to talk about nerve physiology first, and then we get into muscle physiology, because in order to contract a muscle, you have to have a signal to tell it to contract. So it's the same mechanics at a neuromuscular junction. That's the nerve to a muscle. But right now in this unit, we're talking about a nerve onto a nerve, because a nerve goes to another nerve that transmits down to a motor neuron that goes to the muscle that tells the muscle to do something. And guess what? In muscle physiology, you think we're going to move ions on the membrane of a muscle to make it do stuff? Yup. It's the same template. Okay? So if I do this box right here and I blow this box up, that's what we're looking at, is we're looking along the axon and we're looking at how this signal moves from the left side of the screen to the right side of the screen. If it's in green, it's resting, or what we call excitable. And that's corresponding to a slightly negative charge inside the cell. You see at the membrane surface, slightly negative, and then slightly positive on the outside of the cell. And that's in the green zone, excitable. If you're in the red zone where you've got the arrow, so you're starting here, it's moving this way, the arrow is moving this way, action potentials in progress, that's in the absolute refractory period. You can see the inside of the cell is positive. Inside of the cell is positive. Right behind it is this yellow area that's slightly negative, but it's refractory. And that propagates the signal this way rather than it making a U-turn and going in the backwards direction. And that's what I was trying to explain with the toothpaste analogy, but maybe this is a better way to do it. You with me? So our nerve signals here are a traveling wave of excitation. Electrical excitation as it moves. It's a wave front. We can actually do this on a bare axon, unmyelinated. And that's what we refer to as continuous conduction, where we're highlighting a bare, unmyelinated axon. And all of this activity has to happen at the surface. All these channels have to open, and ions have to flow stepwise. Now, if I cover a portion of this axon with myelin, I insulate it, I get what's called saltatory conduction, where the area that the myelin covers, there are no channels that open or close. So the signal, most textbooks in the videos actually show it hopping from one node of Ranvier to the other, but it actually kind of hops on the inside of the cell, not the outside of the cell. The signal moves into the axoplasm and comes up underneath the myelin at the next location. And I'll try to show that here in a second. So myelinated sheaths or myelinated neurons are much faster at conducting a signal than unmyelinated. They get to skip many of the work. Two things that influence the conduction speed: myelination and then the diameter. The larger the diameter of the neuron, the faster it travels. So if we look at this graph, we can see fiber diameter along the x-axis, and we can see conduction velocity in meters per second across the y-axis. And hopefully you can appreciate that myelinated fibers at any given size, like a 10 micron or a 20 micron diameter fiber is significantly faster when you myelinate it. So let me show this video on continuous versus saltatory conduction.
Alternating conduction. Conduction through myelinated nerve fibers. During myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier, which are
Ranvier.
myelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltating conduction. And it serves as a means of increasing the rate of propagation of an action potential.
So, does that make sense? This area that's covered and wrapped by myelin, there's no voltage-gated sodium channels exposed to the outside environment. Essentially, they're covered. Now, they're not even really covered. They're just not made by the cell and inserted there. They're made by the cell and they're inserted in the bare zones, nodes of Ranvier. Right? Or as she says, Ranvier. And so you get ion exchange happening here and then ion exchange happening here. And the signal, it actually jumps inside the cell as it moves here and stimulates the next section that can actually be voltage-gated sodium channels opening. Okay? Let me back up. I think, there we go.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. This multimedia presentation will be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on...
We're not watching the rest. Okay. So I'm using these short little clip videos. Some are pretty old, but I want to show these concepts in different formats for all of you. Okay? So, again, bless you, kind of driving home this concept of why does it propagate only in one direction? Just another way to look at it. So this illustration is showing this axon, and then you've got an area of myelin wrapping, and you have bare nodes of Ranvier. And this sort of explosion right here isn't like the axon just sneezed or something. This is where ions are moving, and they're only moving at these nodes of Ranvier. And so as they move, and this shows it propagating inside the cell, here we've got the action potential in progress. You can see the two negative charges outside the cell, positive charges inside the cell, and it's colored red. And that hops from here to here to here. Again, behind it is the refractory zone in yellow. and all of the membrane in front of it's green, which is excitable or resting. Just a different figure from a different textbook saying some of the same stuff. So let's talk a little bit about multiple sclerosis. And this topic will come back to after spring break. This is a demyelination disease, unfortunately. And it's an autoimmune disease that usually surfaces in young adults, where your body actually attacks the myelin sheath and destroys it. And so you demyelinate neurons. Some of the complications are lack of muscle control. You can ultimately become blind because those neurons that carry vision are absent or deteriorate. And we'll talk about it here in the figures to the right in a second. But this immune system actually utilizes cells known as CD4 and CD8 T lymphocytes. And they harbor an attack against your own tissue. So it's an autoimmune disorder. It is progressively worse as time goes on. It is debilitating. You can live a long time with MS, but you need to likely alter your lifestyle. And many of you will be taking care of patients or treating patients that have multiple sclerosis. So if you can appreciate this top figure, we've got our myelin shown here in kind of the dark blue structures, and then in betweens of the nodes of Ranvier, and they're showing the sodium voltage-gated channels in these sort of like orange inserted transmembrane proteins. So what I want to show you over time as this disease progresses, you lose this myelin, and the way the body makes up for it is it starts upregulating the insertion of new sodium voltage-gated channels in the location where the myelin was. Because now it's bare, it needs to have activity, so over time you lose myelin and you actually move from saltatory conduction to continuous conduction. So in the early phases of the disease, your action potential is just slow, much slower, because you're losing your myelin. Eventually, the neuron actually deteriorates altogether. This is a Nature paper over on the far right. And what I want to highlight is normal myelin, upper right. You've got sort of these kind of white ghost appearances here, and then you've got a bright green, a bright red, a bright green, and then the white ghost appearance. So on either side of this triad structure is myelin. And the red in the middle is a stain for voltage-gated sodium channels. And these green structures on either side are just chaperone proteins that hold it in the membrane. And as the disease progresses from A through D, this was a Nature paper in 2006 out of a neuroscience journal. You see more of the red showing up as the disease progresses. So if you look at the bottom where this arrow is, it's showing a whole string of red, new voltage-gated channels under immunohistochemistry. So the body's trying to compensate. And that's why the disease could be a long-term disease in patients if they're able to upregulate voltage-gated channels inserting into the membrane to compensate for the fact that the myelin disappeared. It's kind of interesting how it works. So we talked a little bit about factors that influence the speed of conduction. Axon diameter was one that I had mentioned earlier. So I talked about myelin and I talked about diameter. So the larger the diameter, the faster the speed of transmission. And the less resistance to current flow with a larger diameter means that you get faster speed of conduction. So in an unmyelinated neuron, you actually have to quadruple the diameter of the fiber in order to double the speed of the transmission. So we would be walking around with really fat axons in our brain if we didn't have the ability to myelinate. But because we can myelinate, we have kind of reasonably sized heads. And when you do brain dissection in 201 lab, you've got white matter and gray matter. The white matter that you're going to see is myelinated neurons. The gray matter is unmyelinated neurons. It's kind of cool. So what happens when the signal now gets to the end of the synaptic knob? It travels down the axon, gets to the end of the synaptic knob at that synaptic button that we showed in the last lecture, that image. Let's talk about some of the terminology. Well, we know we got to get across this gap, which we call a synapse. We've got an electrical synapse and we've got a chemical synapse. The electrical synapse happens where you've got gap junctions that join cells together. So you see an electrical synapse, for example, in cardiac muscle. From one cardiac myocyte next to the other, we had those intercalated discs. And those were linking one cell to another electrically. And that would be an electrical synapse example. A chemical synapse is what we're talking about here with neurons, where we've got the opportunity to use a neurotransmitter. Remember in lecture 1 when we talked about the cocaine example, the video on the cocaine example? So if you haven't watched it yet, now maybe you're interested. You're like, you talked about cocaine? Yeah, we did. We used that as an example at the pleasure centers being stimulated again and again and again, and how cocaine blocks the uptake or the reuptake of acetylcholine that's being released into the synapse. So at the chemical synapse, we've got the opportunity to use a whole variety of different neurotransmitters. And these neurotransmitters have different types of activity or different types of functions. And ultimately, you've got your electrical signal coming down, the presynaptic side. You see all these mitochondria. This is a little cutout where you got all these mitochondria. That's just to prove to you this is just a specialized cell. It's got all the same intracellular components in cell biology that you remember. It's got a nucleus, right? It's got a nucleoli. It has cytoskeletal elements. It has mitochondria because it needs to manufacture ATP. And it makes, through transcription and then translation, it makes proteins and chemicals that it packages in these vesicles that exocytose, fuse with the membrane, dump out their contents into this gap, this chemical synapse, bind to its receptor on the other side of the postsynaptic neuron, and that triggers a voltage-gated sodium channel to open on this side. So you go from an electrical signal to a chemical signal back to an electrical signal. You with me? So let's look at these neurotransmitters. Well, there's a lot of options. In fact, in our body, we've got over 100 known neurotransmitters, and these are chemicals that do what? They cross the synaptic cleft. They can either be excitatory or they can be inhibitory, these neurotransmitters. We've got different categories. We've got acetylcholine in a class all on its own. We've got a variety of different amino acids like GABA, glycine, aspartic acid, and glutamic acid. We've got monoamines like catecholamines, epinephrine, norepinephrine, dopamine. The dopamine was one that we kind of highlighted in the lecture with cocaine addiction. Serotonin and histamine. And then we have neuropeptides like enkephalins, substance P. This one's used for pain. Cholecystokinin and endorphins. This is the endorphin high that you get like on extreme exercise, like runner's high. So all of these either have an inhibitory or an excitatory opportunity, and we're using the same architecture. We're just utilizing different neurotransmitters at different points in the signal. Now, last but not least, and I'll have one final question for you before we wrap up. How do these neurotransmitters work? So they're either in two different categories. Acetylcholine and a number of others are what we refer to as ionotropic. Ionotropic means that they bind on the postsynaptic side and they stimulate ions to move, channels, gates to open. They alter membrane potential. Ionotropic. Think ions. The rest of them are metabotropic. And a characteristic of metabotropic is it utilizes a second messenger inside the cell, like cyclic AMP, if you remember back to basic bio. So the idea here in this figure that I'm showing you, 1, 2, 3, 4, 5, 6, 7, is with cyclic AMP, you've got the receptor seeing a ligand, utilizing cyclic AMP as a second messenger, actually by cleaving through adenosine triphosphate, ultimately making it into the nucleus and triggering the nucleus to go through some sort of gene transcription. So the metabotropic take longer. They're a little slower to occur. More steps than just, hey, I got an electrical gate or an electrical voltage gate. I open it up, ions move. Very simple. Okay? Ionotropic versus metabotropic. Okay, I had a lot of questions for you today. Lecture 3, you're going to see there's a lot more questions. Tried to make it as interactive as possible. All right? It's just going to be you and me on the screen or whatever. But you can pause it. You can replay it. And it's trying to reinforce the steps of the action potential, because this material is the stuff that students tend to struggle with the most. So during saltatory conduction, action potentials jump from where? One astrocyte to another, an axon to a dendrite, one internode to another, one node of Ranvier to another, or one microglial cell to another. D, one node of Ranvier to another. Very good. Any final questions before we finish out today? Yes, sir. Oh, come up afterwards. Questions about ALS?